So thank you so much for having me. It's really a pleasure to be speaking at the 40N scientific webinar. So I am focused on pancreatic ductal adenocarcinoma, which is the most common histologic subtype of pancreatic cancer, and it's the third leading cause of cancer-related death in the United States. And the kind of specific clinical problem I'm focused on is distant recurrence after resection. So this is focused on the lucky few PDAC patients who are diagnosed at an early stage. So these patients can potentially be cured with surgical resection, yet the majority actually have relapsed at a distant site. This most commonly involves the liver, and when it spreads to the liver, that portends an especially poor prognosis. So my long-term goal is to develop novel therapies that block distant recurrence in the liver. But however, right now we're kind of limited in our ability to develop these therapies by an incomplete understanding of the underlying mechanisms. So here I'm illustrating what's referred to as the metastasis cascade. It's the series of steps a primary tumor cell needs to undergo to successfully form a distant metastasis. So it needs to invade the underlying basement membrane, intravasate into a blood vessel, survive in the circulation, extravasate at a distant site, survive the initial interactions with the metastatic microenvironment, including the resident immune cells, and successfully proliferate to form an overt macrometastasis. Um, and you'll see here this dashed line. I've divided this process into kind of pre-dissemination and post-dissemination phases. Only the post-dissemination phases are kind of relevant to this post-resection setting where the primary tumor has been removed. And I'm calling these phases collectively colonization. And that's where we're focusing on in this study. So there have been a number of factors that have shown to contribute to metastatic potential in PDAC, like KRAS allelic imbalance, MYC amplification, EMT, but it's unclear how these contribute to liver colonization specifically. In this project, we're focused on the relationship between cell state or the transcriptional state of the cell and metastatic potential because we think this kind of multiple genetic and epigenetic drivers of cancer, they kind of become integrated into cell state. And so we developed a new mouse model for studying this using DNA barcodes. So the way this model works is I take mouse pancreatic cancer cells, introduce barcodes. So each cell is labeled with a unique repertoire of barcodes. Sort them into single cells, expand these single cells into monoclonal lines, and mix them together equally and co-inject these mixtures into mice. And we can inject these into the splenic circulation, the spleen, and they travel through the portal circulation and seed the liver and form liver metastases. Importantly, these cells come from a pure black 6 background, so they can be transplanted into a syngeneic immune-competent mouse. So this is an immune-competent model. Then I harvest the resulting metastases, sequence the barcodes present in the metastases, and by sequencing the barcodes, I can tell which subclones from the starting population contributed to the metastases that develop. And so here I'm going to show the data from these assays. So on the left, I'm just showing some representative histology and gross pathology of these liver mets, showing that they do kind of recapitulate human disease in the mouse. Here, what I'm showing is histograms of number of clones per tumor for 2 independent experiments with subclones derived from independent primary tumors that I'm calling KPC1 and KPC2. The take-home message here is that most of the tumors are polyclonal, average clonality of 2. And here are the actual results of these in vivo metastasis competition assays. So what I'm showing here, again, the results are broken down into the 2 independent experiments, KPC1 on the left, KPC2 on the right. On the x-axis is the subclone identity, and the y-axis is the fraction of metastases in which that subclone is found. Each dot here is a mouse. The bars are the mean average across mice. And so what you can see is that the subclones in these different populations, they have a range of metastatic potentials. Most of the subclones do not contribute to many metastases, and then a subset contribute to the majority of the metastases. So based on this, we kind of arbitrarily divided the subclones into what we're calling metastasis high and metastasis low, and those are labeled in red and blue. So next, we were interested in uh, determining whether this is specific to liver, and so excuse me, there. So I repeated this process in peritoneum. So pancreatic cancer also frequently spreads to the peritoneum, and I can model this by injecting these same barcoded mixtures directly into the peritoneum of, again, immune-competent mice. They form peritoneal metastases, and I do the same barcode analysis. Again, here I'm showing just representative images of the gross pathology and histology. These resemble human peritoneal metastases. These peritoneal mets, again, are polyclonal for the most part. And when we look at the metastatic colonization potential in peritoneum, what we see is the pattern across the subclones is very similar. So we think the met high subclones we've captured here have a generalized advantage in metastatic colonization rather than a tropism to a specific specific metastatic site. Okay. So now we've captured subclones with high and low metastatic potential. The next thing we want to do is understand, are there epigenomic differences between these that can help explain their differences in metastatic potential? So to examine that question, we performed RNA-seq and ATAC-seq to characterize their transcriptome and their chromatin landscape. And the reason we did this is we figured that the genes and gene programs that enable their metastasis will likely be active at the time of injection. So we performed RNA-seq on the cells in vitro to characterize their pre-injection state. And we also figured that the genes and gene programs that are likely to contribute to this enhanced metastatic potential, which we know is mitotically heritable, are likely to be under stable epigenetic regulation. So we performed a ATAC-seq to characterize open chromatin regions. And then we integrated these two data sets, and that's what I'm showing here. So I'll walk you through that. So each dot here is an open chromatin region or peak identified in a ATAC-seq. These are differentially accessible peaks between met high and met low. So the x-axis is differential accessibility between met high and met low, with more open towards in met high towards the right, more open in met low towards the left. And then we linked each peak to the nearest gene, and the position on the y-axis is differential expression of that gene. So higher expression in met high towards the top, higher expression in met low towards the bottom. And you can see that most of these dots are black, and meaning that the difference in expression is not significant between met high and met low. And that isn't unexpected because many of these, some of these genes may be in a poised state. Some of these peaks may be regulating a gene other than the nearest gene. Some of these peaks may not be regulating any gene's expression. But when we look at peaks linked to genes with significantly differential expression, they follow the expected pattern. So 97% of the time, if the peak is more open in met high, the gene has higher expression in met high. 91% of the time, if the peak is more open in met low, the gene has higher expression in met low. And we use this concordance between gene expression and chromatin accessibility to define what we're calling the met high genes, and there are 207 of those, and the met low genes, and there's 182 of those. So of course, next we wanted to try to understand, what is the biological significance of these genes? What are they doing? So we applied gene set enrichment analysis to the met high genes, and we see enrichment of pathways involved in inflammation, like TNF signaling pathway that I'm highlighting here. When we look at the met low genes and again perform gene set enrichment analysis, we see pathways related to tissue development and differentiation, motility, and Wnt. I'm highlighting here the genes that are currently identified in these different pathways, and this is a heat map showing their expression across the different subclones. The met high subclones here are towards the right and met low on the left. So this I think what this is illustrating and kind of our take-home message from this is there's a complex dysregulation between met high and met low, but there are kind of two kind of clear features. We see activation of inflammation-related genes in MET high. So specifically pro-inflammatory cytokines, pro-inflammatory cytokine receptors, and the kind of machinery for converting extrinsic signals into NF-kappa B activity. And then in MET low, we see activation of transcription factors involved in islet development. So pancreas has two main compartments, exocrine and endocrine, the islets of Langerhans. So pancreatic cancer is thought to develop in the exocrine compartment. But here we're seeing in the MET low subclones activation of transcription factors that drive neuroendocrine development. The motility and Wnt related genes are harder to interpret because they don't kind of point to a single clear phenotype. So I think the best we can say is just there is a dysregulation in motility and Wnt between these two states that we need to investigate further. Consistent with our pathway enrichment, we also performed transcription factor footprinting, which is a technique very specific to ATAC-seq data to identify transcription factors with increased activity in MET high state or MET low state. And in the MET high state, we see increased activity of all five of the NF-kappa B transcription factor subunits, which I'm showing here highlighted in red. So that's consistent with the enrichment of the inflammation-related pathways in MET high. And we see enrichment of a HOX transcription factor, HOX A13, and a para-HOX transcription factor, CDX2, in MET low. So these are involved in anterior-posterior patterning during development. And I think this is, again, kind of consistent with activation of pathways that are typically seen during embryonic development in the MET low subclone specifically. So we now have identified genes and gene pathways that differentiate the MET high and MET low subclones. The next thing we want to do is understand how these two different states fit into the evolution of PDAC. And so the chromatin landscape, chromatin dynamics during PDAC development have recently been mapped. And this was done in a paper by Alonso Corbello and colleagues in 2021. And so we wanted to see how our subclones fit into this evolution of PDAC. And so I obtained the data from this paper, which involves harvesting epithelial cells from different mouse models, normal pancreas, inflamed pancreas, pre-neoplasia plus inflammation, and then full-blown PDAC. And then they performed bulk ATAC-seq on all these different samples. And so I obtained their data, I processed it in the same way as my data, did a batch correction, and plotted all of the samples together in the same PCA space, and that's what I'm showing here. So what you can see is that I was able to recapitulate what the authors showed in their paper, which is that these samples arrange themselves along a normal to PDAC trajectory based on ATAC-seq data, with normal pancreas here towards the left. This is pre-neoplasia inflammation plus pre-neoplasia, and then PDAC is on the right. And then my samples fall within the PDAC region here, as expected. But if we zoom in on my samples here, MET high and MET low are the filled and hollow shapes. You can see that these don't separate along this normal to PDAC axis. These are intermingled with each other and basically have the same position along the normal to PDAC axis. So our take-home message from this is this metastatic potential axis that we've identified is independent of the normal to PDAC trajectory. In line with that, we also looked at the MET high genes and MET low genes, their differential expression between PDAC and normal pancreas. Again, using data from this Alonso Corbello et al. 2021 study. And what we see is that the majority of both the MET high and the MET low genes are increased in PDAC relative to normal pancreas, and the relative ratio is not different between these two groups. So again, this speaks to the orthogonality of the two axes, and I think this is what it's telling us is that MET high and MET low are differentiated by differential activation of pro-tumor pathways. So conclusions from this first part of the talk: we have identified a novel cell state axis associated with metastatic colonization potential of PDAC subclones. And this is met high and met low are differentiated by a number of complex features, but the two that seem to be the most clear are activation of inflammation-related genes in met high and activation of gene expression programs specific to adjacent developmental lineages in met low. And our model for how these develop is that these subclones arise from distinct branches during PDAC tumorigenesis that diverge along this metastatic potential axis, which is orthogonal to the normal to PDAC axis. And also, I haven't mentioned this, but a classical basal axis. These are two molecular subtypes that are have been established in PDAC. Okay, so we've identified a metastatic potential axis in a mouse model. Of course, next we wanted to assess its relevance to human disease. And so to do this, I obtained bulk transcriptomic data from human PDAC data sets that have both primary tumors and metastases. And to assess their position along this axis, we have developed a single sample scoring metric that we call MET score. This is built upon a computational method called SING score, which basically is a rank-based method that scores a bulk transcriptomic sample based on enrichment of an upset and simultaneously depletion of a downset. So here we defined the upset as the MET high genes and the downset as the MET low genes. And this version of SING score we're calling MET score. So I'm going to walk you through the data. In these two human PDAC data sets, what we see is the metastases have higher MET scores than primary tumors significantly. And in agreement with this finding, when I looked at a PDAC single-cell RNA-seq atlas and score all of the single cells for MET score, which I'm showing here, and break these down based on their donor source, whether these cells came from a metastasis or a primary tumor, the metastatic cells had higher MET scores than primary tumor cells. This is really important because this rules out stromal contributions to MET score in the bulk RNA-seq and bulk microarray samples. So this basically what this is showing is that this metastatic potential axis we discovered in a mouse model, it appears to be conserved in human PDAC. And further supporting this, primary tumor MET score in PDAC is prognostic of overall survival. So here I'm looking at three independent human PDAC data sets. In each, and these are now just early stage patients with localized PDAC. When I stratify the patients based on MET score, just top half of MET score versus bottom half of MET score, patients with a high MET score have a worse overall survival in each cohort. And these are called, I know this audience is diverse. These are called Kaplan-Meier curves, where we're basically showing the fraction of surviving patients over time. And this is summarized in a forest plot on the right. So next, what we wanted to look at is, so it seems like this metastatic potential axis is conserved between mouse and human PDAC. The steps a cell needs to go through to metastasize are similar between different cancer subtypes. So we figured that this axis may be conserved in other cancer subtypes. And so what I'm showing here is indeed in these independent cohorts of colon cancer, breast cancer, and prostate cancer, across all of these, we see higher MET scores in the metastases relative to primary tumors, suggesting that this metastatic potential axis is indeed conserved amongst cancer subtypes. These are all carcinomas, so these are all derived from epithelium. This is very intriguing. When I look at a melanoma cohort, the pattern is reversed. So the metastases have lower MET scores than primary tumors. Melanoma is derived from neural crest. So we think that what this is telling us is that this metastatic potential axis is shared amongst carcinomas, but not broadly shared with non-epithelial malignancies like melanoma. Similarly, when I look at primary tumor MET score in mismatch repair proficient colorectal cancer or hormone receptor positive breast invasive ductal carcinoma, high MET score is prognostic of worse overall survival in both of these cohorts. And this is the final slide in this section. I think this is very important. So We also looked at whether MET score may be predictive in colorectal cancers, since this seems to perform very well in colorectal cancer. And for the non-physicians in the audience, prognostic means the score tells us something about the likely natural course of disease. Predictive means the score will tell us something about how the disease will respond to therapy. And predictive is going to be a predictive biomarker is much more impactful than a prognostic biomarker, because a predictive biomarker will change the course of therapy. And so in early stage colorectal cancer, uh, a current challenge is whether or not to give chemotherapy after resection of that tumor and how much chemotherapy to give. And what I'm showing here is that the benefit of adjuvant therapy or postoperative therapy in stage 3 mismatch repair proficient colorectal cancer, there's a significant benefit to postoperative therapy for the patients with high MET scores, but there is not for patients with low MET scores, suggesting that MET score could be a predictive biomarker that could guide these decisions in localized colorectal cancer. So some conclusions from this part of the talk. So our cell state axis identified in the mouse model appears to be conserved in human PDAC and also across multiple other human carcinomas. And primary tumor MET score is associated with overall survival in multiple human carcinomas and is predictive of adjuvant therapy benefit in mismatch repair proficient colorectal cancer, suggesting that this could have utility as a clinical biomarker. And that's something we're going to be assessing in the future. Final thing I want to talk about today is, we've identified the set of genes that's enriched in PDAC subclones with high metastatic potential. We hypothesize that this set of genes is likely to be enriched with genes that are functional drivers of liver colonization. So these are genes that, if we deplete them genetically or if we inhibit them therapeutically, these could reduce the colonization potential of these subclones. And so to test this, we did kind of a proof of concept where we did a targeted, small-scale targeted shRNA screen focused on inflammation-related genes, since we saw that these genes were enriched in MET high. And this is kind of the scheme of the screen. So we introduced an shRNA library into a MET high PDAC subclone. We generated liver metastases, again, by splenic injection. And as our control, we generated primary tumors with orthotopic injection. We used this control to subtract out any effects related to proliferation or engraftment. And then we sequenced the shRNA loci in the metastases and the primary tumors, basically to look for enrichment or depletion of these shRNAs over time in these two sites. And I'm showing the data here. Each dot here is a gene targeted by multiple shRNAs. So it's basically showing the average effect of all the shRNAs targeting that gene. Position on the x-axis is log2 fold change of shRNA abundance, higher in primary towards the right, higher in liver metastases towards the left. And on the y-axis is our statistical significance with our alpha cutoff highlighted by this horizontal dashed line. And so we have 4 hits in the screen. And where these shRNAs targeting these genes are depleted in liver metastases relative to primary tumors, suggesting that they are positive functional drivers of liver colonization. We focused on our top hit FOS for further validation. And to validate this further, we took an independent MET high PDAC subclone, generated knockdown lines, and co-injected the knockdown line with a non-targeting shRNA line. And again, for these 2 different shRNAs, we see depletion of the FOS targeting shRNA in liver metastases. And with SH-FOS number 1, this is statistically significant. With SH-FOS number 2, there's a strong trend, likely limited by the kind of modest degree of knockdown in that line. And knockdown of FOS, importantly, doesn't diminish in vitro proliferation of these lines. So we don't think we're capturing something kind of trivial like just a generic effect on cellular fitness. We think this is a specific effect on liver colonization. So take home from this part of the talk, CFOS, the transcription factor CFOS, encoded by FOS, is a positive functional mediator of PDAC liver colonization across multiple tested MET high subclones. And we therefore think this is an attractive therapeutic target in the adjuvant setting. Um, some exciting future directions, I think, for building on this work, exploring MET score as a predictive biomarker for adjuvant chemotherapy benefit in localized colorectal cancer, exploring CFOS as a therapeutic target in localized PDAC for intercepting metastatic recurrence. And going forward, I want to perform a comprehensive screen of all 207 MET high genes. And I'm thinking right now of integrating this with a perturb-seq readout and machine learning to predict gene combinations with the greatest synergistic effect when co-inhibited. I'd like to acknowledge people that help with the work and also funding sources. And as mentioned earlier, I'm going to be starting my own lab at Emory. We're going to be hiring later this summer, so keep an eye out for that. And if anyone's interested in learning more about the work, I have a link here to a preprint, and we hope that the manuscript will be coming out soon. Thank you so much. Happy to take any questions.